DNA is made out of multiple nucleotides. Each nucleotide consists of a phosphate, a sugar, and a base. The first carbon attaches to the base. The second carbon attaches to a hydroxy group if it's RNA or a hydrogen if it's DNA. The third carbon bonds to the next 5' phosphate, and the fifth carbon attaches to a phosphate. DNA is made up of 4 different rings: cytosine, thymine, adenine, and guanine. Cytosine and thymine are pyrimidines, and guanine and adenine are purines. Pyrimidines are composed of 1 ring, and purines are composed of 2 rings. Nucleotides attach to each other through phosphodiester bonds, meaning the phosphate that is attached to the 5' carbon of one nucleotide will attach to the 3' carbon of another. This creates the phosphate backbone, which is what makes DNA slightly negative. Hydrophobic interactions between stacked base pairs helps to stabilize DNA. DNA strands are antiparallel, meaning that the 5' end of one strand lines up with the 3' end of another strand. DNA also consists of a major and minor groove. As seen on this DNA, this is the major groove and this is the minor groove. And these grooves alternate. DNA helixes are usually right-handed, meaning they have a clockwise spiral. As seen here, the phosphate backbone points to the right. You can also see it in this yarn that also points to the right. In a high salt concentration, DNA becomes more stable because the negatively charged phosphates become shielded. This then eliminates the repulsive force between the 2 antiparallel strands. Since the DNA is more stable, the DNA will be less likely to denature when heated. Therefore, the DNA will have a higher melting temp, or TM. This is like when you put your hair up in a ponytail. The hair is pulled back but still loose. If you add hairspray, then there's more of a connection and the hair is tighter.